Assalamu alaikum everyone. Hope you all are doing well. First of all, I would like to introduce myself. I'm Anila Khan and today I'm here to present my topic which is on terms of biotechnology. As we know that there are many terms of biotechnology but I will mention a few here. So let's move to the first term of biotechnology. This is immobilized enzyme. An immobilized enzyme is an enzyme attached to an inert insoluble material such as calcium alinate. This can provide increased resistance to changes in conditions such as pH or temperature. As we can see that in the picture, here are some chemicals and physical methods of immunized enzymes. The next term is immunoassay. An immunoassay is a biochemical test that measures the presence or concentration of macromolecules or a small molecule in a solution through the use of antibody or an antigen. The next one is intellectual property. Intellectual property is a category of property that includes intangible creations of the human intellect. There are many types of intellectual property and some countries recognize more than others. The most well-known types are copyright, patents, trademarks, and trade secrets. Here is the diagram of copyright machine. You can see guys. Here is the landfill. A landfill site also known as a tip dump, rubbish dump or dumping ground. It is the site for the disposal of waste material. There are many methods of landfill and landfilling is the term used to describe the process by which solid waste is placed in the landfill. A few terms are described in this slide guys. Here is the lignocellulose. Lignocellulose refers to plant dry matter, so-called lignocellulosic biomass. It is the most abundantly available raw material on the earth for the production of biofuels, mainly bioethanol. The second one is metabolite. A metabolite is an intermediate or end product of metabolism. The term metabolite is usually used for small molecules. As in the picture, you can see that primary metabolites and secondary metabolites, these are the two types of metabolite. The third one is microarray. A DNA microarray is a collection of microscope DNA spots attached to a solid surface. Scientists use DNA microarrays to measure the expression levels of large number of genes. The next one is micropropagation. Micropropagation is the practice of rapidly multiplying stock plant material to produce many progeny plant using modern plant tissue culture methods. The next one is monoclonal antibody. A monoclonal antibody is an antibody made by cloning a unique white blood cells. All subsequent antibodies derived this way trace back to a unique parent cell. The next one is multipotent cells. Multipotent cells are cells that have the capacity to self-renew by dividing and to develop into multiple specialized cells. As you guys see in the picture, there is one cell and dividing it into three cells and also the three cells are dividing into multiple cells. So this process is called multipotent cells. The next one is mutagen. A mutagen is a physical or chemical agent that changes the genetic material, usually DNA of an organism. The next term is mutation. Mutation means change. A mutation is a change in a DNA sequence. Mutation can result from the DNA copying mistakes made during cell division, exposure to ionizing radiations, exposure to chemicals, exposure to mutagens, or infection by viruses. Here is the in the diagram there is two template of DNA. The first one is the original sequence, and the second one is point mutation occur because the a change in the DNA sequences occur. The few terms are described in this slide. This one is mycelium. Mycelium is the vegetative part of a fungus or fungus-like bacterial colony consisting of a mass of branching thread-like hyphae. The next one is mycoprotein. Mycoprotein is a form of single-cell protein also known as fungal protein. Protein derived from fungi, especially as product for human consumption. The next is patent. A government authority conferring a right or title for a set period. It is like a license given by the government. And this is phenotype, the set of observable characteristics of an individual resulting from the interaction of its genotype with the environment. And last one is plasmid. A plasmid is a small extra chromosomal DNA molecule within a cell that is physically separated from chromosomal DNA and can replicate independently. It is the last slide of my topic. This one is pluripotent cells. Pluripotent stem cells are cells that have the capacity to self-renew by dividing and to develop into the three primary germ cell layers. The last one term is PCR. PCR is called polymerase chain reaction. It is the method widely used to rapidly make millions of copies of specific DNA sample. It allows scientists to take a very small sample of DNA and amplify it to a large enough amount to study in details.